Hi student, and this video lecture we will talk about one of the important topic from the animal diversity, that is about the body wall of sponges, or you can say the integumentary system of the sponge. But before discussing the body wall of the sponge, we should have a sound knowledge about the two terms, that is what is diploblastic animal, and the second one, that is what is triploblastic animal. Now you can see here the embryonic development. So from the fertilization or from the gametogenesis, when the gametes are formed, mean the sperm and ova is formed. When these sperm and ova they are combined with each other, they will form a product that is called as zygote. Now and this zygote, the further developments occur or the division occur. Now next stage is form and the developmental process, that is called as cleavage. Now in this cleavage, morula stage is occur and blastula that is formed after the cleavage. Now in this blastula, another step occur, that is called as gastrulation. Now at the end of this gastrulation, the embryo form two layer, or in some organism the three layer is present, or two or three germ layer is present. Now from these two or three germ layers, the whole body of the organism is formed. Now at this stage, we will divide the animal of the kingdom Animalia into the two category, that is called as diploblastic animal and triploblastic animal. Actually, the layer that is called as ectoderm, the second one that is endoderm, while the layer that is found between these two layers, that is called as mesoderm. Now we will divide these three layer, actually represent these two terms, that is diploblastic and triploblastic. So the animal and which two layer is present, that is the term diplo is used, mean two layers. So only the these two layer is present in the organism, and the whole body of the organism that is formed from these two layer. So such type of the organism that is called as diploblastic animal. Now look at see the definition, that the body of the organism formed from two germ layers. So actually these germ layer are, actually these organisms are called as diploblastic animal. Now look at see what are the two germ layer that is present in these diploblastic animals. So these are actually the ectoderm, while the second one, that is endoderm. Actually these two layer that is responsible for forming the body of these animals. Now actually the layer that is found between the ectoderm and endoderm, that is called as mesoderm. So in case of the diploblastic animal, the mesoderm layer, it is actually absent. Now the example of this diploblastic animal, that is sponges, mean phylum Porifera and the phylum Cenetrate are nidarian. So these two phylum actually are diploblastic animal. The body of these animal that are from the ectoderm and endoderm, while the mesoderm that is absent in these organism. Now you can see here the a non-gelatinous, a non-cellular genitalious layer it's found between the ectoderm and endoderm in this organism. So these gelatinous matrix are non-cellular layer that is found between the ectoderm and endoderm and this organism, that is called as mesenchyme, or you can say the mesohyl. So these are actually the gelatinous matrix that is found between the ectoderm and mesoderm in case of the Porifera or sponges, while in case of the coelacanth, these gelatinous matrix that is found between the ectoderm and endoderm, that is called as mesoglia. So you should have to remember the name of these two gelatinous matrix. In case of the spongy, it is mesohyl or mesenchyme, while in case of the coelacanth, this is called as mesoglia. Now the second term, that is about the triploblastic animal. So the body of this organism that is developed from three germ layers. Now these animal are called as triploblastic animal. As the name indicates, triplo means three. So in this organism, the three layer that is found in their embryo. So from these three germ layer, the whole body of the organism that is formed. Now what's the name of these three germ layers? These three germ layer are actually the ectoderm, endoderm, and the third one, that is mesoderms. So the animal, the example of the phylum and the kingdom Animalia, that is actually the organism from the platyhelminths to the chordates. So all these phylum are the organism that is found in this phylum that are actually the triploblastic animal. Their body contain these three germ layers from which the whole of the body that is formed. So this is about the diploblastic and triploblastic animal. Actually, by understanding the body wall of the spongy, we should have the information about these two terms. Now let's see the body wall of the sponges. As we know that the sponges are actually the diploblastic animal. So the animal that's their body that is mainly formed from the two germ layers. So such type of the animal that is called as sponges. But here we will discuss the body wall of this sponges. So these are not actually the germ layers. So you can see here these are not the germ layers. So the true diploblastic that contain two germ layer and their embryonic stage, that is actually the Cnidarian. So Cnidarian is actually the true diploblastic animal, that you should have to remember this point. Now, if we look to the body of the sponges are for reference, so mainly two cellular layer is found in the body of the sponges are for reference. Now, the outermost cellular layer of their body that is called as pinacoderm, while the innermost cellular layer of the sponge body that is called as choanoderms. Now, and between these pinacoderm and choanoderm, a gelatinous matrix is found. Now, this gelatinous matrix that is called as mesenchyme, or you can say the mesohyl. So it is actually a gelatinous matrix that is found between the pinacoderm and choanoderm. Now let we discuss each layer in detail. So the first one, that is pinacoderm. Now first of all, look at see the etymology of the term pinacoderm. Now this pinacoderm, it is merely derived from the two Greek words, that is phanex, while the second one, that is term. Now the phanex, it means that the dish, while the term it is, and biology the term that is, this is used for the skin or layer. Now why it is called as dish layer? As we look the structure, as we look the structure of the pinacoderm, it is mainly comprised by a well-nominated structure that is found or still found in this pinacoderm, that is called as pinacocyte. Now this pinacocyte, it is mainly continuously combined with each other and form a continuous sheet which give a plate-like structure to this pinacoderm. So on this look like a flat structure, this layer, it is called as dash layer. So this is about a short intro about the meaning of pinacoderm. Now look at see the definition of the pinacoderm. So this is actually the outermost cellular layer of the sponge body. This is called as pinacoderms. Now this pinacoderm, we have two type of the pinacoderm. This is called as exopinacoderm, while the second one, that is endopinacoderms. Now what is exopinacoderm? So this is actually cover the outermost surface of the pinacoderm. So the pinacoderm, which is mainly exposed to the environment or outer surface. So this layer, it is called as exopinacoderm. As this exo, it is used for the outside. Now, this exopinacoderm, it covers the entire body of the sponges. So this is only or the outer, the entire body of the sponge. This is comprises by the exopinacoderm. So mainly at the osculum and dermal ostia, this exopinacoderm, it is absent. Now at the osculum and the dermal ostia and the epithelial lining of the incurrent canals, another layer of the pinacoderm that is found, that is called as endopinacoderm. So this endopinacoderm, it is towards the layer of the pinacoderm which is toward the inside. This is called as endopinacoderm, as this term endo mean inside. So this endopinacoderm, it is mainly include the epithelial lining of the incurrent canal. So this endopinacoderm, it is mainly present at the epithelial lining of the incurrent canal. So this is mainly the canal that is mainly responsible and the canal system of the sponges. So here the endopinacoderm, this is also found in the osculum and dermal osseous. So this is about the location of these endopinacoderms. Now look at see the general structure of the sponge body. Now this is the overall morphology of the sponge body. As you can see here, a small tiny holes is present over the all body of the sponge. Now these tiny holes are pores that is present in the sponge body, that is called as ostia. So through this ostia, the water that is enter into the sponge body. Now you can see here a large of the opening is present that is called as osculum, through which the water leave the sponge body. Now here a structure that is present at the base, this is called as through which this spongy it is attached with a subprotein. Now this attachment structure, this is called as base. Now this is overall the general morphology of the sponge body. Now we will take a cross section of this sponge body. Now you can see here that the body wall, this consists of two cellular layer that is shown in this figure. Now you can easily clearly see this structure. So this is actually the base through which it is attached with the substratum. Now you can see here a tiny outer cellular layer that is called as Pinacoderm. Now you can see here this is the Pinacoderm which contain epinacocyte cells. So this is the pinacocyte cell. Now this is exopinacoderm, while here at the lining of the incurrent canal, this is actually the endopinacoderms. While this yellow color, it is actually show the ostia through which the water entered through the body of the sponge. This is actually a large opening. Now this large opening, this is called as osculum. So you can easily here see that this is a gelatinous layer that is actually the mesenchyme or mesohyl. So you can see here here at the right at the middle a cavity is present. Now this cavity, this is called as spongiocele. At the center, a large cavity that is found at the sponge body are present, that is called as spongiocele. Now right in the spongiocele, a flagellated cell is our funnel shaped cell that is found. So this flagellated cell, this is called as choanocytes, and this innermost layer, this is actually called as choanoderm. So this is pinacoderm outermost, while here a flagellated cell that is present at the spongiocele sides. Now this is called as choanoderm. So this is about the structure of the sponge bodies. Now, a well-nominated structure are the cell that is found in the pinacoderm that is called as pinacocytes. Now, look at the definition. Right at the endopinacoderms contain a large, flattened polygonal cell that is called as pinacocytes. As you can see this structure of this pinacocyte, so this is the structure of pinacocytes. Now at the center it is a large, so it is flattened structure, while it contain a polygonal cell, mean it contain a many sites. So you can see here a many site is present in these cell. So this is called as polygonal cells. So this is actually the definition of pinacocytes. Now the second point that is regarding to the structure of this pinacocyte. So in shape the pinacocyte possesses a center bulging contain the nucleus. So at the center you can see here, and the membrane of these cells are epigenetes are moved outside. So this is actually a center bulging that is found, and this bulging contain a nucleus. Now, the margin of the adjacent cell are closely cemented together, as we have already discussed in the etymology of the pinacoderms. So, as I have already discussed in this the etymology of pinacoderms, that the cell pinacocyte cell and the pinacoderm layers, these are connected with each other and form a flat-like structure. So, the margin of these cells, they are connected with each other. So, this is mainly the mean of these centrines. Now, what is the function of these pinacocyte cells? So these pinacocyte cells are actually highly contractile and can greatly increase or reduce or decrease the surface area of sponge body. Mean these cells are mainly responsible to increase or decrease the surface area of the sponge body. This is mainly the function of this pinacocyte cell, a well-nominated cell that is found in the end, especially in the end of pinacoderm. It is also present in the exopinacoderm. Now discuss the modification and that occur in this pinacocyte cell. Now actually this pinacocyte cell at the various region and the sponge body are in the pinacoderm sites. They will modify it and form a specialized type of cell. Now from this pinacocyte it will modify it and form mainly two type of the cell. The first one that is porocyte, while the second one that is myocytes. Now the first one that is porocytes. So what is porocyte? It is actually the pinacocyte in the lining of incurrent canal. So this some pinacocyte are modified to form a tubular cell. So this type of cell, this is called as porocytes. So mainly at the incurrent canal, this pinacocyte, this is modified and form mainly the tubular cell. Now this pinacocyte, it is now called as porocytes. Now let's talk about the characteristics of these porocytes. This is actually a thin walled cell which is mainly often at the both ends, that have a nucleus which is present at the periphery of the cytoplasm. So this is mainly the three characteristics of this porocyte. The first one, that is it is thin wall. The second one, that is both end of this cell it is often, while the nucleus that is present at the site of the cytoplasm, that is at the periphery sites. Now, what is the function of this porocyte cell? So this porocyte cell, it is mainly often at the bone end. So mainly this porocyte cell, it play important role in the connection of the radial canal and the incurrent canal. So mainly it is connecting the incurrent canal with the radial canal through a structure that is called as prosopyle. So now this prosopyle contain the porocyte cell which mainly connect the incurrent canal with the radial canal. So this incurrent canal and radial canal that we will discuss in the canal system of the reference and details. Now the second cell that is formed from the modification of pinacocyte, that is called as myocytes. Now mainly it's the region of the osculum, dermal ostia and inner ostia. These pinacocyte cells that are elongated, contractile and act as a muscle cell. Now this pinacocyte cell, this is now called as myocytes. So what is the function of this myocyte cell? Now this myocyte cell, this is mainly occur at the opening of the this structure. Now this opening, this small opening, this is called as ostia, while this large opening through which the water leave the sponge body, this is called as osculum. Now mainly this at this region, the pinacocyte, it is modified and form as muscle like cell, this is called as myocyte. So which is mainly responsible for to regulate the movement of waters. So this is mainly the function of this myocyte cell. It regulates the movement of water. Now the second cellular layer of the sponge body, that is called as Choanoderm. Now, first of all, look at me, see the meaning of the Choanoderm. Now, this term is actually derived from the two Greek words. The first one, that is con, which actually mean that the funnel, while the term it means the skin. Now, what does it mean, funnel layers? So, actually, here the important characteristic cell that is found in this layer, that is called as choanocytes. Now, this choanocyte have a funnel-like shapes. So, that's why this layer is called as choanoderms. Now, what is the Choanoderm? Specifically, we define the Choanoderm. So, it is actually the innermost layer of the sponge body is called as Choanoderms. Now, this Choanoderm, it's actually from the gastric epithelium. Now, here in the Choanoderms, the choanocyte cell is present, so which is mainly responsible for the digestion of the foods. So, partly the digestion that is mainly occur in the sponge body. So, partly the partial digestion that is occur in the choanocyte cell, while the partial digestion that is mainly occur in the spongiostele cavity of the sponge body. So it mainly from the gastric epithelium. Now the main diagnostics cell that is found in this choanoderm layer, that is called as choanocytes. So this choanocyte, it is also called as flagellated cell because it bear a flagella. So that's why it is called as flagellated cell. Now, the presence of choanocyte is the characteristics of the phylum Porifera. Now, this phylum Porifera have two important diagnostic feature or characteristics. The first one, that is the presence of canal system, while the second one, that is choanocyte. So these are the two important characteristics that is not found in other phylums. So the main recognizable feature of these two phylum Porifera, that is presence of canal system, while the second one, that is choanocyte cell. Now look at see the structure of the choanocyte cell. Now this choanocyte cell contain a single nucleus, one or two contractile vacuoles, food vacuoles, and a single basal structure that is called as kinetosome. Now from this kinetosome, the flagella, a web-like structure arises that is called as flagella. Now look at see this is the structure of atypical choanocyte cells. Now this choanocyte cell contain here a large nucleus, a single nucleus is present. Now this is the nucleus, while this is the cytoplasm of the choanocyte cell. Now here the food vacuole is present, while this one it is actually the black spot. It represents the contractile vacuole that is present in the cytoplasm of this choanocyte cell. Now here the important structure it is present at this. Now from this structure, a specific web-like structure, that is flagella, which arises from this structure, it is called as kinetosome, which help in the arrangement of the flagella. Now here the this component it is present. Now this component it is called as collar region. So on the presence of this collar region, this conocyte cell, it is called as collar cells. Now what is the function of this important conocyte cell? So here the one point that is left that we will discuss, and then we will finally discuss the function of the conocyte cell. Now flagella is surrounded at base by a thin cytoplasm collars. So you can see here this is the base of the flagella. So here a thin cytoplasm it is present. Now this thin cytoplasm it is called as collars. Now what is the function of this conocyte cell? So mainly this conocyte, the first function of the conocyte cell, it contain a flagella. This flagella it is responsible for beating. Now this when this flagella it is beats, so the water it can easily enter to the body of this sponges. So this is the first function. Now after the entering of the water molecule or water, so it carry a food itself. Now this conocyte cell it is mainly responsible for the digestion of food. So these are the important two functions of this conocyte cell. Now the other part that is found in the body of the sponge, that is called as mesenchyme. Now this mesenchyme it is very important part that is found between the Pinacoderm and Choanoderm layers. Now this mesenchyme it is also known as mesohyles, while in case of the coelenterates this layer it is called as mesoglia. Now what is the proper definition of mesenchyme? So it is actually a gelatinous matrix that is found between the Pinacoderm and Choanoderm. This is called as mesenchyme. Now this mesenchyme it contains an amoeba-like cell that is called as amoebocyte cells. Now this mesenchyme it actually contains a cell, that cell is it is called as amoebocyte cell. Now amoebocytes are modified arcucyte cells that migrate from outer layers and carry on many functions. So actually at the outer layers, a special type of cell it is present that is called as arcucyte cell. Now this arcucyte cell it is modified and form this amoebocyte cell, so which perform various type of the function. Now on the basis of the function, this amoebocyte cell it is have the following types. Now the type of the amoebocyte cell. So here are the important type of these amoebocyte cell. On the basis of their function, we have the six amoebocyte cell. The first one that is chromocyte cell, second one that is collencyte cell, the third one that is arcucyte cell. So arcucyte cell it is actually the early or embryonic amoebocyte cell. The fourth one that is scleroblast, while the fifth one that is schizocytes, while the sixth one that is gland cell. So let's talk about shortly one sentence about this type of the amoebocyte cell. Now the first type of the amoebocyte cell, that is chromocytes. As the name indicates, chromo means colors. So these are actually the pigmented amoebocyte cell, which is mainly responsible to give color to the sponge body. As the sponge body have different type of the colors, so this color is just mainly given by the cell that is present in the sponge body, that is actually the chromocytes. Now the second one, that is collencyte cell. So this is actually mainly the function of this collencyte cell that bind the outer and inner layers of the sponge body. So it acts as a binding molecules. Now the third one, that is arcucyte cell. So arcucyte cell it is very important. So this is actually the embryonic amoebocyte cell and can be differentiated into others any other cells. So it have the ability can be converted into other type of the cells. So that's why it is called as totipotent cell. So totipotent cell are actually cells which can be converted into any other type of the cells. So the which of the following are actually the totipotent cell among these amoebocyte cells? So actually the arcucyte it is a totipotent cells. So you have to remember this for MCQ's point of views. Now the fourth one, that is scleroblast cell. So it is actually the type of the amoebocyte cell which is mainly responsible for the formation of the endoskeleton of the sponge, that is called as spicules. On the basis of the nature of the spicule, we have the various type, that is calcioblast, silicoblast and spongioblast. So these are the various type of this scleroblast cell. Now the fifth one, that is schizocyte cell. So these are actually responsible act as a storage cell, while the last and sixth one, that is gland cell. So gland cell, it is mainly, gland is actually the group of cell or cell which is responsible for the secretion. So mainly these cell are secrete an adhesive substance, because these are actually sessile animals. So at the base, a specific type of the cell it present, that is called as gland cell. Now this gland cell secrete an adhesive substances. So the main purpose of this adhesive substance, that it provide amine for the attachment for the animal. So the for the attachment of the animal with a substrate, they should have a cell that is called as gland cell, which secrete an adhesive substance. So this is all about the body wall of this fungi or integumentary system of this fungi body. Thank you so much.